Hello, everybody. Welcome to this Labor Day lab makeup narration. This is one way to keep pace with the rest of the sections in the semester. And what you see here is pretty much what you would have seen through a Zoom session had you attended this lab that we all miss. I hope you had a great Labor Day. But here is the next segment of our uh, hierarchy in these living systems. From the cells, we're moving to a segment where like-minded cells group together to form tissues. So we are mentioning a huge number of functions that individual cells fulfill. They can just do this more efficiently with higher power, with bigger volume, if these cells group together. So consider the function of neurons, although we won't talk about them today. But if you want to create any kind of conscious thoughts, just one cell of the nervous system, one neuron is not enough. You need billions of them. Therefore, we have to have nervous tissue. We talked about energy storage through triglycerides in the chemical basis of life chapter. Cells that store fat, adipocytes, by themselves could store a minuscule amount of this energy carrier. If we have adipose tissue, now we have millions of adipocytes storing pounds and pounds and pounds of this energy carrier. So we have an advantage coming from like-minded cells grouped together. Tissues weren't known for a long time to biology until microscopy have developed. And even then, the early microscopes could show very little when you stuck a piece of tissue under that microscope. Most human tissue is translucent to light, so thick chunks of tissue show up as a blur. And even if you would manage to slice the tissue very thin, just on its own, a tissue would show a transparent image, not too many features would be seen. So in the 1800s, there was a revolution in many parts of the world, and the textile dyes led to much more colorful clothing, and some of those organic dyes were discovered to increase the contrast of visible structures on tissue, biological material. So this is why I want to show what exactly we'll be looking at today. If we had face-to-face labs, you would be looking through the microscopes, and it's I think just good to know what goes into creating a tissue slide. The actual tissue is right here, this purple rectangle. For a tissue to be to show us any visible structures, first the cells need to be killed, and it's done with some kind of alcohol or aldehyde. Then the tissue needs to be sliced extremely thin, almost like hair thin. And then that slice needs to be stained. So the wonderful colors, mostly hues of pink today that comes to us, are not the natural colors of this tissue. They are artificial. They are added to the tissue during processing. So it's good to know that what we are looking at is man-made colors to increase visibility of microscopic structures. Once the tissue slice is stained, then it is put on a glass slide. A thinner cover slip glass is placed on it, and it's glued at the edges, and you have a preserved human slice of tissue, stained, ready for viewing, and those are the slides we'll be looking at today. Now, when histologists looked at the human body first, they were expecting thousands of different tissue shapes and arrangements, because there are thousands of ways our body functions. Each organ system has several organs. A number of different functions can be assigned. So the expectation was that we're going to have hundreds, if not thousands, of tissues. To their surprise, not only that they saw a limited number of shapes and arrangements, but after a while, all these tissues could be put into 4 separate categories. So this is a very simple arrangement that our body keeps using and reusing again. So those 4 tissue categories are the epithelial, connective tissues, those 2 topics we cover today. And in next week's lab, you'll be seeing the nervous and muscle tissues. So those are the 4 categories. Epithelial tissues is our first topic, and under the microscope, epithelial tissues show visually different arrangements. So in general, epithelial tissues are a set of cells that form a fairly thin sheet, and by thin, think about saran wrap or similar thinness. The cells can be in a layer of single cells, and this is called a simple epithelium. Sometimes we see 2, 3, sometimes more layers added onto each other. These are called stratified epithelial tissues. And then, based on the shape of the cells that make up that thin layer, we have yet another set of names. As you can see, this is all highly, highly visual, just a testament to what can be actually observed through the microscope. So when we have flat tile-like cells touching each other to form this sheet, the name for that epithelium is squamous. When we have square, cube-shaped cells forming this layer again, see the cells are all touching each other. This is the cuboidal type of epithelium. And then we have epithelial cells that look like columns, and they are yet again touching each other at their side, and these are the columnar epithelia. In the tissue chapter in the lecture, we'll talk about just what exactly holding these cells together, gluing these cells together. We're not going to get into that in lab. In general, the function that can be fulfilled by the variety of very thin layer of cell is usually covering body surface, lining the internal surface, internal lumen of structures, all organ systems that communicate with the outside world, respiratory, digestive, urinary, reproductive systems, they all have the lining, internal lining, made up by epithelial tissues of one sort or another, and we'll see quite a few examples. Even the inside of every blood vessel is lined by an epithelial tissue, and this will be the simple squamous. So as we go through these categories, we will be looking at a combination of these names, like simple squamous, simple cuboidal, stratified squamous, and there will be 2 epithelial tissues out of the 6 that have colloquial names. Here's our first example, simple squamous epithelium, and this is good to say a few words about these, what I call tissue sheets or tissue pages. These are in your lab shell. Not only that you see an image that you're supposed to recognize and identify, name the tissue category, but in terms of questions on graded assignments, you might be asked about tissue category, you might be asked about location in the body and function. The typical features may come up. I may mention that when that's an important aspect of the tissue identity or functioning. So simple squamous epithelium, this is the group of cells that form flat sheets. The tile-like cells are not readily visible cell by cell, but look at these thin structures. This is an image from our lung, and the air sacs, the alveoli, are lined by simple squamous epithelium. So here is one alveolus, and I'm just following the outline of it. Here's another one. In artistic rendition, alveoli are nice, round, cup-like structures. After processing, those cup-like structures are crushed a little bit. But here you see another air space. Here's another one. Here's another one. And the lining of it is all these very thin, simple squamous epithelium. This is the simple squamous epithelium that also lining blood vessels. The function of this is to promote diffusion. Gases, especially in the alveoli, move through these cells in and out of the blood, so promoting diffusion. In the respiratory system chapter in AP2, we'll see just how concentration gradients drive diffusion, but I'm really hoping that after the cell lab, the word diffusion rings a bell. So this is the type of tissue where diffusion is promoted by the very thin separation between air and blood in the alveoli. The simple cuboidal epithelium, here we're looking at hundreds of these tubes, as you can see these, from our kidneys. This is where it's good to take a look at where the actual epithelial cells are. And when you look at these round, darker purple structures, those are the nuclei of the cell. So imagine a sheet that is curled up into a tube, into a straw, and we are looking at hundreds of those straws on this image. In our kidneys, we have about 1 million of these straws, and huge amount of mostly active transport is taking place through them. So the function here is secretion and absorption. Again, in AMP2, we'll be going into great details. Just imagine every day 47.5 gallons of fluid is moving through the cells that make up these kidney tubules. Humongously efficient part of waste product removal in our urinary system. So again, the cells are forming the walls of these straws of these kidney tubules. Please notice diffusion is not mentioned here. These cells are too thick to efficiently promote diffusion. The simple columnar epithelium, this is one of the group of cells that are really tough to visualize. We are here in the small intestine, and in our small intestine there are these long finger-shaped structures, the villi, that reach into the nutrient-rich liquid that's moving through here. So this is the lumen of our small intestine. The cells, maybe this is where you can see the most, and I'm sorry I can't zoom in on these images, but you see column-like cells here. This is probably the best place. In the small intestine and also in the large intestine, there is a lot of interruption of this nice regularly placed columnar cells, And the interruption comes from these pearl looking bright, round looking cells. These are called the goblet cells, the named here under typical feature. You are expected to recognize these cells. Their function is to make mucus, therefore contribute to the lubrication in the small and large intestine. Function of the simple columnar epithelium is absorption and secretion. And in our small intestine, in the digestive system, AMP2, we'll come back to these structures, these cells, and identify just how active and passive processes absorb nutrients and other materials from the lumen of the small intestine. Here is yet another simple epithelium, although name would make you believe that this is something different. A long time ago, this epithelium was thought to be stratified, but then it turned out that despite the appearance of the epithelium, it's fake stratified. It's still considered simple epithelium. We are looking at a cross-section of the trachea here, and the epithelium is this purple line here. This lower right corner of the image is the lumen of the trachea, and so this is the pseudostratified ciliated columnar epithelium. There are other tissue types here, and many connective tissues are found to support this layer. So a large portion of the slide actually is not showing this epithelium, only this line of purple cells is the pseudostratified ciliated columnar epithelium. Here too we see goblet cells, a little less obvious. I'm going to point to this oval bright area here. Here's another one. Here is the third one. So goblet cells produce mucus here. So trachea, bronchi is a location. The name ciliated indicates that there is a fine line of bristle-like structures. Again, you would have to zoom in, and maybe you can do this on your device. You would see these bristle-like structures. Those are the cilia. And the secretion function comes from goblet cells. The protection function comes from the movement of these cilia on top of the cells. They are clearing our airways of debris, pathogens that get trapped in the mucus. So cilia are these actively moving portions of these pseudostratified ciliated columnar epithelial cells. Our first stratified epithelium is a stratified squamous epithelium. So the squamous shaped cells are on top, and these very thin thread-like cells are the squamous cells. The rest of the epithelium is appearing in this dark pink color. If you can zoom in, you can see 1, 2, 3, 4, 5, 6 cells on top of each other, so definitely stratified. And here, no transport function. Secretion or absorption is listed here. The function of these fairly thick epithelial layers is protection. Take a look at the locations. Every place there's an expectation of some physical stress. So lining of our oral cavity, outer layer of the skin, lining of the vagina, anal canal. So, and we could add to this the esophagus. Every place stress on the epithelium is expected, and so protection, the thick layers allow the depth or the rubbing off of a few top layers, but epithelium is still there to separate the lumen of these organs from the rest of the tissues. And the last epithelial tissue that we're going to look at here in the lab is yet another stratified epithelium. The name doesn't quite say that. It's called transitional, reflecting the very important function of this epithelium, which allows stretching to occur without breaking the epithelial. So in this image, the epithelium is very thick, purple layer. What you see here is the lumen of the urinary bladder. So urine would be collecting in this space. As the bladder fills, the bladder needs to stretch, and the epithelium needs to stretch with it without breaking. We don't want to allow any content of the urine to seep into the underlying tissue. This wedge-shaped tissue is rich in blood flow, so we don't want content of the urine to reach this portion of the body. So this sealing layer needs to hold. And here the cells are present in so many layers. They hold onto each other so incredibly well that when you stretch the bladder, the cells may turn into a very flat shape, but they still hold. And so allowing stretch is the most distinguished function of the transitional epithelium. Lining of the urinary bladder or ureter also lined with this tissue. The second category that we'll be talking about today is the connective tissues. So in epithelial cells, epithelial tissues, the expectation is the cells hold onto each other. They form a solid, preferably unbreakable layer for whether it's allowing diffusion or secretion, absorption, or for protection. Connective tissues look strikingly different. Here, the cells are separated from each other, no longer this gluing together, and the separation is done by large volume of this mostly proteins and polysaccharides, collectively called matrix. Sorry for this duplication here. So this colorful threads and some diffuse material is called a matrix. So connective tissues are nothing else but a combination of cells plus matrix. As you'll see, in these tissues, we are going to pay attention to just what is in the matrix, because that has a decisive effect on the function of the tissues. Connective tissues is the category that shows us a tissue that is liquid. Blood is a connective tissue, and what makes that tissue liquid is the matrix, which we call plasma. Bone is a connective tissue, very different from blood, right? We are expecting bones to hold up under our weight. Think about your tibia holding up almost the entire weight of the body. We cannot have a liquid connective tissue making a bone. So the matrix in the bones have minerals and proteins coming together to form this incredibly solid, hard tissue resistant to pressure. We'll see connective tissues where flexibility is achieved by just adjusting the components of the matrix. So while cells function, but in connective tissue, the matrix has a very important effect on the overall function of these tissues. The fibers influence them perhaps most, and in the connective tissues where we do see, or at least where we do know that fibers are functionally important, we mention these fibers. Collagen, elastic, and reticular fibers are the types. Collagen fibers tend to be very thick and resist physical forces. We'll be seeing them and mentioning them in those tissues that have that function. Elastic fibers allow flexibility in tissues in a way that you can deform those tissues and yet they will regain their shape because of the elastic fibers. Think about elastic fibers as a protein fibers that act similar to rubber. No matter how you stretch it, you crumple it, it still regains its original shape. Reticular fibers have just a minor supporting function. In a lecture about tissues, we'll say a lot more about elastic and reticular fibers. Here in the lab, we are limited to the microscope slides we actually have in the lab. So let's take a look at this list of connective tissues. The poster child for connective tissues, and this is good to start with the areolar connective tissue. Here the cells are these purple clumps, and you can see that they are definitely separated. And what's in between them is sometimes this kind of blotchy purplish material, but sometimes you can see, looks like hairs. These are the fibers, so these are all made up of proteins, sometimes polysaccharides. And so this is a typical appearance of connective tissue. And you can see the cells are no longer attached to each other to form a sheet. Here we have to mention that we have all three fiber types present: collagen, elastic, and reticular fibers, and it's listed here under typical feature. We have to emphasize the role of the fibroblast. The fibroblasts, and you might want to take a note of this. If you're following your lab report is asking more detailed questions about fiber type, cells that are creating the fibers. So please take a note or notes on that, or you can just take a note on a separate piece of paper. But fibroblasts are the cells that actually make the matrix. Adipocytes, macrophages, mast cells are just guests in the areolar connective tissue. Some of them stay for a while, some of them just move through. Areolar connective tissue is the most abundant connective tissue because it is under all epithelial types. There is a specific mention here, papillary region under the epidermis. I put this here because in the last lab exercise we'll be talking about a set of models, microscope slides of the integumentary system, and we will point out the papillary region under the epidermis, and that is made up of areolar connective tissue. What is the function of areolar connective tissue? It provides strength. Think about collagen fiber, elasticity through the elastic fiber, and support through the reticular fibers, so that thin saran wrap like epithelium receives some kind of a support that makes those epithelial layers a little more sturdy. So this is areolar connective tissue. Our second connective tissue is adipose tissue. Here we're not going to say much about the matrix. The cells function to store triglycerides and fats, and that determines the function of the entire tissue. The cells are showing up as these large circles. However, the center of the cells, these light-colored ovals, represent triglyceride storage spaces. So the cell, like the organelles, nucleus, the Golgi apparatus, the ribosomes, are all squished into a thin layer of the cytoplasm around. The matrix is very sparse. You can see a little bit thicker appearance of the matrix here. Again, matrix does not have a decisive role here. So Important to write down the name of these cells. These are the adipocytes or fat cells. In the previous tissue we mentioned fibroblasts. So cell names will be a part of the typical features that are important to note. You may be asked on a graded assignment, name the cell a pointer is pointing out, and in the adipose tissue, the only cell we are concerned about is the adipocyte. So it's natural adipose tissue functioning as storing fat as an energy reserve. It also can protect organs. We have adipose tissue around heart. Kidneys have a generous layer of adipose tissue. And we also have the thermal insulation function. This is important mostly of the adipose tissue under our skin. Insulation from extremes of the environmental temperatures, in part done by a layer of adipose tissue. The next three tissues here will be three different versions of cartilage. These are hard connective tissues. We'll be looking at cells that need to find a place where they are protected from all that pressure. So hyaline cartilage is our first cartilage. One of the locations, and maybe functionally most noticeable location, is at the end of long bones. It's really important if this is the location you're putting on your flashcard or highlighting that you use this entire term. We have many bones in the body that are not long bones. Think about the bones of the skull. You don't see much hyaline cartilage at the end of those. But at the end of a long bone, such as a femur, or the humerus, or the tibia, and many others, we do see hyaline cartilage. There the function is to provide a smooth surface to reduce friction. Our joints move a lot easier with hyaline cartilage put or positioned at the end of the long bones. We also have respiratory systems such as trachea to keeping our airways open. So the C-shaped cartilage rings of our trachea are made up of hyaline cartilage. When you take a closer look, most of this tissue is made up by this pink, very smooth looking featureless area. This is all matrix. Important to know that this is rich in collagen fibers, providing strength to this tissue. The cells that make this matrix and maintain it are these small oval-shaped cells. These oval-shaped cells are called chondrocytes. This is in the typical feature, and they reside in microscopic cavities, and those cavities are called lacunae. When you look at some of these ovals, take a look at this one. There are two cells here, and during processing the cells shriveled a little bit, so you can see this white space showing you the outline of the cave, the cavity, the lacuna. The cells need to reside in caves because think about what happens. We have hyaline cartilage on the distal surface of our femur, on the proximal surface of the tibia, namely the knee joint, right? So when you stand up, most of your body weight is pushing down on this hyaline cartilage. Your body weight would squish these cells, no problem. We can't lose these cells because they maintain this cartilage. So this location and cavity provides them some protection from whether you're standing up, walking stairs, or jumping. So the cells find nice residences in these lacunae. Elastic cartilage is the only tissue where we are going to name elastic fibers as the dominating element. So the expectation is that this cartilage can be deformed and will be regaining its shape. And indeed, our external ear is made up under that skin area by elastic cartilage, and you can crumple your ear no matter how badly you can sleep on it for hours, yet your ear will regain its shape in the morning. We have an elastic cartilage in our throat, epiglottis. In AMP2 we'll come back to that. This cartilage has to bend during swallowing to prevent food and drinks getting into our airways. We also have elastic cartilage around our nostrils. When you blow your nose, you squish those nostrils shut, and when you let go, your nostrils regain their shape. So there's a thin ring of elastic cartilage in that part of our body. The general makeup of the elastic cartilage, general tissue design, is repeated for all three cartilages. We have chondrocytes, and you can see one chondrocyte here, you can see two over here, and the lacuna is outlined by this darker brownish dark purple material. So you can see quite a few of these clear ovals, and those are the cavities, the lacunae. So just like with hyaline cartilage, elastic cartilage is also based on chondrocytes hiding in caves in the lacuna. Our last cartilage is fibrocartilage, very different color. You can see the wavy, very visible nature of the thick collagen fibers. Those are the blue ones. Chondrocytes are very sparse. You can see one here, another one, 3, 4, maybe 5, 6 here. The general design here, yet again, chondrocytes, those tiny blue, tiny purple dots in the lacuna, which is the larger clear ovals. The location for fibrocartilage are the intervertebral discs, these cartilage discs located between our vertebrae. Pubic symphysis also has this material. Provides strength and rigidity to joint structures. If you notice, fibrocartilage is always joining bony structures that move only a little bit. So when you stand up, your vertebrae are supporting the shape of our torso. The fibrocartilage has to live up to and be strong enough up to the rigidity and material strength of the bone that's below and above these slices of cartilage between our vertebrae. Here's this world famous bone tissue. We do have several structures here. This is surprisingly the most organized connective tissue. Inside our bone, at least in one type of bone, we see these large arrangements, this large oval, and you can see part of the second one. Here's the third, here's the fourth one. These large oval units are called osteons. And the center of the osteon is the central canal. The osteocytes, the cells that fill the lacunae here, are in these dark, thin oval areas. We cannot observe the osteocytes, but their cavity, the lacunae, are right here. And you can kind of make up several layers of the mineralized matrix. So here's one layer, here's the second, Here's the third layer, and each layer is called a lamella. So this is the makeup of the unit, the osteon. Location is in bones. So, the function is not only supports, protects, think about the cranial bones protecting our brain, and of course all bones store minerals because of the large amount of mineral matrix that is located between the osteocytes. Our last connective tissue is the blood. Blood is this connective tissue where the matrix has a lot of water, some proteins in there, but the matrix itself is liquid and cells are suspended in this liquid matrix. So we have a liquid tissue that is able to transport and fulfill other functions. So let's take a look at the individual components. Most of the cells called red blood cells. The red blood cells appear more like pinkish, maybe light brown, depends on, I guess, how the screen is showing them. You can see tiny circles. These are all red blood cells. The function of the red blood cells is oxygen transport, and it's noted right here during the function. The white blood cells appear as purple entities here, so we see quite a few. White blood cells have immune protection function. There are there are 5 types of white blood cells that are swimming in our circulation. In AMP2, in the cardiovascular chapter, we will detail those. Right now, all expectation is that you can name these purple cells as white blood cells. And then we have the plasma, this lightest colored background in between all these cells. The plasma has probably the biggest transport function. Please notice plasma transports carbon dioxide. The red blood cells have a relatively small capacity to transport carbon dioxide. Overwhelming majority of carbon dioxide is transported in the plasma. The plasma is the matrix responsible to transport nutrients. So when we talk about blood sugar is transported in the plasma, all amino acid. What about those good and bad cholesterols, LDL, HDL, that transports lipids and cholesterols? They're all swimming in a plasma. What about these antibodies? We are hoping to protect us from the COVID-19 disease once immunization comes about. Antibodies are also in the plasma. Notice we didn't mention one part. In lecture we will mention platelets. Platelets are not really visible, especially at this magnification, and so for purpose of practicality we'll leave them out of our consideration. In AMP2, in the blood chapter, we will say a lot about platelets. They do have important functions, but they're not readily visible on this particular slide. If you look at your lab report, on the last page, there are several questions about what tissues, epithelial and connective tissues would you use to construct a respiratory, urinary, or cardiovascular system. And this question relates to this new development, a decade or two old development in science called tissue engineering, where tissues, sometimes entire organs, can be constructed by cells that artificially put together into layers. So think about what would you do if you're a tissue engineer and if you have to construct a respiratory system. What are the tissues would you use from the ones that we covered here today? Epithelial, connective tissues. because this is the toughest thing to do in a narration, to ask everybody to focus on one or another particular line of that lab report. I usually do this by asking students to provide the answer. So for you guys, please write a list of these tissues, what you would choose to construct those organ systems, and the organ systems are mentioned in the location part of the slides, the tissue slides. So this is the end of this narration. When you're listening to it, please let me know if you have any questions. Don't hesitate to message or email me. And please don't forget, you do have lab home assignment about this lab. The tissues will feature in a major fashion in your practical, so it's a really good idea to practice recognition and location, function, tissue categories, cell types, fiber types on these tissues for the lab home assignment. And we will have a lab quiz as it's scheduled on our schedule. So I'm going to stop recording here. And again, if you have any questions, please don't hesitate to contact me.